Hello students, welcome to Daily Chronicles by Abhiram Ias. So today let's discuss the important articles from the various papers. Okay, the topics I've selected are Nalanda and Vikramshila University and also about the SC Collegium, and we'll also discuss about DNA polymorphism and also Manregas, and the other two articles are from PIB, where the articles are National Mission on Culture Mapping and also about Project Pari, and last article is Meghalaya's Leaving Route Marble. Chalo, let's start the articles. This first article is very important for the exam, so please do listen carefully. And the first article tells that after Nalanda, another Bihar university now set to rise from the ruins. That another university is Vikramshila University. Okay? Now, let's see what the article is telling about. So, basically the article is telling, a decade after Nalanda University came up in the foothills of Rajgir, work is on to revive another ancient center learning in Bihar, that is Vikramshila University. Now, Amma, here what is significance of Rajgir, which is located in Bihar? Rajgir, which is located in Bihar. So, the significance is it served as the capital city for the Mauryan dynasty. Okay, Mauryan dynasty. And also for the Haryanka dynasty. Okay. Who are the important rulers in Haryanka dynasty? Bimbisara, right? Bimbisara and also Ajatashatru, right? Bimbisara and Ajatashatru. That is the significance of Rajgir. Now, in this context, let's see about Nalanda University and also about Vikramshila University. Now, as the article is mentioning about Vikramshila, first let's see about Vikramshila. Now, this is the Vikramshila University pictures, okay? You can see the ruins, okay? Ruins. See the pillars and see how it is structured, right? And this one also. These are the pictures of Vikramshila University. Now, let's see about Vikramshila University. So, if we see the historical significance, it was founded by the Pala king Dharmapala. Now, who has founded this Pala kingdom? Pala dynasty? It is Gopala. Okay? Gopala. Pala kingdom was founded by Gopala, but this Vikramshila University was founded by the Dharmapala. Okay? Now, what is the specialization of this Vikramshila University? It is known for the Tantrayana Buddhism. That means also Vajrayana Buddhism. Okay? Tantrayana or Vajrayana Buddhism, right? Next, the most important scholar who belongs to Vikramshila University is Atisa Deepankara. Okay, remember this? Atisa Deepankara, and in exam they may ask you like this scholar is related to which type of Buddhism. So he is related to mostly Vajrayana Buddhism or Tantrayana Buddhism. Okay, so who is he? The most important scholar belongs to Vikramshila University is Atisa Deepankara. Okay, right, next. Now, how did it decline? So, because of now, how did it decline? It declined because of Bakhtiyar Khilji's invasion. Okay, it declined due to Bakhtiyar Khilji's invasion. So, this is historic significance of Vikramshila University. Next, let's see the key structures of Vikramshila University. So, basically as we saw, it is cruciform stupa where the central structure with 4 staircases. Here you can see 4 staircases, right? So, cruciform stupa with 4 staircases and integrate brickwork. If you see here, it is all made up of bricks, right? Bricks. Now, next, and also some prayer halls. Okay, you can see some monastic cells. Almost 208 monastic cells can be seen, which are for like living quarters for monks, which are arranged in square pattern. Okay, and there is also manuscript hall. That is same like library. Okay, manuscript hall. And also teaching halls and debate chambers, and also temples and shrines dedicated to Buddha and also dedicated to Hindu gods. Okay, like we can see Vishnu, Ganesh temples also, okay, etc. And also there are more fortified walls and gateways, okay, which are protective walls and grand entrances, possibly for like different scholars, okay. Now next, let's see the differences between Nalanda and Vikramshila universities. So Nalanda is founded by the Kumaragupta one from Gupta dynasty, but whereas Vikramshila is founded by the Pala dynasty, who Dharmapala. Right? Now you can see it is established around 427 CE, but here it is in late 8th century or early 9th centuries. Okay, where it is located? Both are located in Bihar itself. Now next, what is the purpose of this? Higher learning and various disciplines. And this is a university. Of course, it is a learning hub, right? And here also, specialized in Buddhist studies, especially the Vajrayana Buddhism. And here mostly Mahayana and Hinayana, okay? Mahayana and Hinayana. Mostly Mahayana and also Mahayana and Hinayana. Who are the notable scholars here? So, Nalanda University notable scholars are Aryabhatta, Zhuge and Atisa. Now, coming to Vikramshila, it is Atisa Deepankara. He covered both. Okay. Now, what are the libraries? Dharmakunj. The term is Dharmakunj, where the mountain of truth, which is a vast collection of manuscripts and texts. Here the library is, it has extensive collection of Buddhist texts. There is no such name, but remember in Nalanda University, Dharmakunj was there, which is a mountain of truth. Many books were there in Nalanda University, which were destroyed. So, now who caused the destruction of Nalanda University? Same, Bakhtiyar Khilji around 1200 CE. Here also destroyed by Bakhtiyar Khilji around 1193 CE. None of these dates, but it is destroyed by Bakhtiyar Khilji. Right? This is all about the Nalanda and Vikramshila universities. Now, moving to the next article. The next article is about SC Collegium. Now, why it is in use? Because SC Collegium recommends transfer of Justice Verma. Let's see the significance of SC Collegium. But before that, let's see why it is in use. So, the move to transfer this Delhi judge to Allahabad High Court comes as an inquiry into allegations of burned currency in his home and is said to commence. So, judge is likely to be excluded from the judicial work in the new post. When he is appointed in the Allahabad High Court, he will be excluded from the judicial works. Now, let's see about the SC Collegium. Now, what does this collegium system means? Simply nothing but this collegium system is the one where it is appointing and transferring the judges in the higher judiciary, like Supreme Courts and High Courts. Okay. Now, see, this is not mentioned in the constitution, Okay. Remember this, but evolved through the judicial interpretations. And further, the President of India appoints the judges based on this recommendations of the collegium. Clear? Now, next. Let's see the composition of Supreme Court Collegium. Now, what does this composition consist of? Definitely CJA, that is Chief Justice of India, and 4 other senior most judges of the Supreme Court. Okay, 4 more senior judges of the Supreme Courts. And now what they does? So they recommends the SC judges appointment, that is Supreme Court judges appointment, and also the High Court judges appointment. Right? Now, let's see the evolution. How does this collegium system has been evolved? There are 3 judges cases. Okay. Now, coming to the first judge case, it is SP Gupta versus Union of India in the year 1981. Now, what is it tells? So, basically the first judge's case was about executive versus judiciary, and the key issue was who has the final say in the judicial appointments, whether it is executive or whether it is judiciary. Now, what was the verdict? So, it has given upper hand for the executive, citing that government has the primacy in judicial appointments, like CJ's opinion is not binding. Okay, it has given upper hand for the executive. Now, what was the impact? It gave more powers to the government, reducing the judicial independence, right? But whereas coming to the second judge's case, what happened? It is also known as Supreme Court Advocates on Record Association versus Union of India in the year 1993. Now, what it is telling that? So, can the CJ's recommendation be binding in appointments? Yes, definitely. So, it overturned the first judge's case and established the collegium system, where CJA plus senior judges have the primacy. In the second judge's case, the collegium was established. Now, what happened? It shifted the powers to the judiciary from the executive. Okay. Now, next is third judge's case, that is the presidential preference in the year 1998. Okay. Now, what happened? So, how should the collegium function? Based on the president. Okay. So, expanded the collegium, where consists of if it is SC appointments, CJA plus 4 senior judges from the Supreme Court, and when it is High Court appointment, like High Court judges appointment, it is CJA plus 2 senior most judges from the respective high courts. Okay, now what happened? It strengthened the judicial primacy, reducing the executive role. Okay, and collegium became the final authority in the appointments. In middle, they have also introduced the NJAC. Okay. Okay, before talking about NJAC, let's see about the procedure for appointment of the judges. Okay, now how are the Supreme Court judges appointed? Basically, collegium recommends the names to the law minister, and the law minister forwards it to the prime minister, who advises the president. Therefore, president appoints the judge. Now, in the High Court judges, so this it consists of High Court collegium, where CJA plus 2 senior most judges recommends the names, and the SC collegium, the Supreme Court collegium, reviews and approves. So, and also law minister, prime minister and president finalizes the appointment. Remember this procedure for appointment of judges. Okay. Now, let's see about NJAC. What is NJAC? National Judicial Appointments Commission, which was a failed reform basically. Now, what does NJAC consist of? So, basically in 2014, they tried to replace this collegium system with a body including members from the judiciary, executive and also from the public. Okay, but it was struck down, citing that it is unconstitutional. Okay, struck down in the year 2015, calling it as unconstitutional. Right, clear about SC collegium? We have seen what is collegium. We have seen the composition of Supreme Court collegium. We have seen the 3 judges case. Next, procedure of appointment of judges, and also lastly about NJAC. Now, why this is why the NJAC was criticized? Because of lack of transparency and lack of judicial supremacy, and also nepotism and favoritism. Okay, so lack of transparency, no clear criteria for the selection; judicial supremacy, no role of executive or legislature. Okay, nepotism, no accountability in the judge selection. Now further, let's see about DNA polymorphism, which is in news. So the article is telling what are DNA polymorphisms and how do they differentiate between the people. This is important in context of science and technology. Okay, many times such questions are asked. Please do read this article carefully. So now, before knowing the DNA polymorphism, let's talk about DNA. What is DNA? Deoxyribonuclease, everyone knows that. Deoxyribonucleic acid, and also it is the hereditary material present in our chromosomes, right? Now, it carries the genetic information that determines the traits of the organism. Now, DNA is made up of nucleotides, each containing a sugar molecule, that is deoxyribose, and a phosphate group, okay? And also the adenine, thymine, cytosine and guanine, okay? A, T, G, C. Now, this DNA is a double stranded, okay? This DNA is a double stranded and follows the base pairing rule, where A pairs with T and G pairs with C, okay? Now, and these are present in the chromosomes, okay? See, A is pairing with T and C is pairing with G, right? Now, next, let's see about DNA polymorphism. What is polymorphism here? See, simply it is nothing but variation of the genes. So, polymorphism refers to, yes, variations in the DNA sequence that occur at the frequency of more than just 1% in a population, okay. Now, it is responsible for the genetic diversity among the individuals, and these variations will be occurring due to 4 causes, but the important cause is mutations, okay, in the DNA sequence, and are passed from one generation to the other generation. Sometimes it may not be also, it may not pass also, okay. And this DNA polymorphism do not usually affect the biological functions because mostly occur in the non-coding regions of the DNA, right? Now, next see some examples of DNA polymorphism. So, there are types of DNA polymorphisms. First type is single nucleotide polymorphism. What is a single nucleotide polymorphism? A change in single nucleotide, okay. If the pair is like ATGC, ATGC, there occurs one change, like the sequence changes to ATGC, ATCC sometimes. So the nucleotide missing here is G, Okay, if it happens like this, the single nucleotide polymorphism. Now, example is sickle cell anemia. Sickle cell anemia, where due to one gene replacement, you will get the sickle cell anemia. Now, so HBB gene mutation, CAG, CAG2, sometimes it will be like CTGC. Here, instead of A, you are seeing T, right? So that is the single nucleotide polymorphism. Now, what is the effect of the sickle cell anemia? Our oxygen absorption will be less in the body. Okay. Now, where it is used in disease suspectability, ancestry, and also the forensic studies, right? Next, coming to the short tandem repeats, which is also called as microsatellites. Okay, short tandem repeats, which is also called as microsatellites, and this is basically used in your fingerprinting. Okay, fingerprinting also, and also paternity testing and genetic profiling also. Okay, short tandem repeats, where 2 to 6 base pair sequences will be there, which will be repeated multiple times. At one point of time, the gene will be replaced. So, TH01 gene, it is repeated like n number of times, repeated used in forensic DNA fingerprinting. Our fingerprint differs from person to person, right? So, this is the sequence. Now, the next step is variable number tandem repeats, which is also called as mini satellites. Why? Because here 10 to 60 base pairs repeating DNA sequence. Here it is only 2 to 6, that is microsatellites. Here it is mini satellites. Why? Some 10 to 60 base pairs will be repeated. Now, what is the example? Insulin gene VNTR, okay, where it is causing type 1 disease risk. Simple. See, the base pairs will be like arranged in this manner, whatever the manner, like n number of times. But at one point of time, there will be a change of nucleotide, like 80 GG. In place of C, you will get G. So due to this replacement of one nucleotide, you are getting all these mutations. Okay, here it is single nucleotide change, but here in the base pair sequence, you will get the nucleotide change, so the entire sequence will be changed, right? So it is for like 2 to 6, but here it is 10 to 60. Okay, now these both are mainly useful for DNA fingerprinting and also population genetics. Now, last is insertions or deletions, which we will call as indels. Okay, where small additions or deletions of the DNA segments itself. So, as I told you, see, ATGC, suppose there is a deletion of the CG, okay, then you will get this indels. Okay, now this is the example for this is cystic fibrosis, okay, where delta F508 mutation in CFTR gene causing cystic fibrosis, okay, and this is mainly used for disease studies, genetic disorders, and also forensic analysis. Clear? Once again, let's see type of DNA polymorphism. So types are single nucleotide polymorphism, short tandem repeats, where it is also called as microsatellites, where only 2 to 6 base pair structures repeats, and variable number tandem repeats, where we call as mini satellites, where 10 to 60 base pair will be repeated, and here it is insertions or deletions, the last type, where small additions or deletions in the DNA segments. Clear? See, remember this and also remember the examples. Clear? Now, next, let's see why it is caused. As one of the primary thing is due to mutations, but there are also other causes. Okay. First is mutations, spontaneous changes in the DNA sequences, and other is recombination, where DNA exchanges during the meiosis cycle. Okay. Next is natural selection, naturally it occurs, and also genetic drift, random changes in the allele frequencies over generation, from as it is passed from one generation to other generation. So that is the genetic drift. So the causes are due to mutations, that is spontaneous changes, recombinations, where during the meiosis cycle, natural selection, it is caused naturally, and genetic drift when it is passed to one generations to other generations. Now, next, what is the significance of these all DNA polymorphism? Yeah, already we saw for forensic science, where fingerprinting, right, paternity testing, right, due to DNA, and also genetic disease researches, that is studies, evolutionary studies also from how we are generated, right? Now the last is pharmacogenomics. What is pharmacogenomics here? It is simply nothing but study of drugs, I mean the effect of genes on how the drug is affecting. Okay, study of genes, I mean how the genes are affecting the drugs. Next, that is all about the DNA polymorphism. Now let's get into the next article. What is next article? It is about Manregas. First, let's see why it is in news. So basically it is in news because the Ministry of Rural Development has issued some guidelines to tackle the challenges of high rate deletions of job cards issued to the workers under the Mahatma Gandhi National Rural Development Guarantee Scheme, that is here Manregas. Okay, what is Manregas? Mahatma Gandhi National Rural Employment Scheme, right? Now let's see the proper news analysis, summary of this. So basically Ministry of Rural Development has issued some guidelines for deletion of job cards, that is Manrega cards. So in past 4 years around 10.43 crore cards were deleted. Okay, now what are these guidelines? So basically he told, first verify if who are not eligible for the Manregas, then please verify and remove their job cards. The next, what are the valid reasons for this? So you have to whether the person is shifted permanently to another place, like permanent migration, and also urban settlement from rural areas. If you have come to the urban areas, so please to remove the Manrega card. And other is duplication of the cards, and other is if the person is dead, please to remove the Manrega card. Okay, now and also he said there should be transparency measures, like if you are removing the card, it should be maintained in the public domain up to 30 days. Okay, then the review should be twice a year. Okay, twice a year you have to review the cards. And the last one is if you are removing card, if there are pending dues, please do clear it, then remove the card, right? This is the summary of the article given. Now let's see about the Manregas. What is this Manrega? It is Mahatma Gandhi National Rural Employment Program under the Ministry of Rural Development, and this is like it is established under Mahatma Gandhi National Rural Employment Guarantee Scheme, which was established in the year 2005. Now next, what is this objective? First is employment, rural employment, livelihood security in rural areas. Next is durable assets like roads, ponds, irrigation facilities, and also empowering the women, like empowering rural women, where jobs will be given for 100 days of work, and also strengthening the decentralized governance through the Gram Panchayats. Now, who gives this Manrega cards? That is Gram Panchayats. Cards is given by central government, but who will tell whether they are eligible or not? That is Gram Panchayat. Now next, what are the challenges in this Manregas? Duplicate cards, and also delayed payments and wage calculation issues, fake cards, technology challenges, that is Aadhaar based payments, as is Manregas is like DBT, direct benefit to your account. Now if you are not linking your Aadhaar, what happens? Your money will not come to you. The next, underfunding leading to work demand suppression. Okay. Now, next article is about National Mission on Culture Mapping and Project Pari. This was there in news couple of times. That is why I selected this article. It is important for the exam point of view. So, the article is National Mission on Culture Mapping and Project Pari. Let's see what the article is telling about. So, it is given the cut here. So basically the article is telling about National Mission on Culture Mapping and Project Pari. Let's see about them. Now what is this National Mission on Culture Mapping? Basically the objective here is simple. Document and promote the India's cultural heritage to revitalize the rural economies. Okay. Now who is the implementing body? Remember this is very important. It is Indira Gandhi National Center for the Arts. Okay. Implementing body for this National Mission on Culture Mapping is Indira Gandhi National Center for the Art. Next, What is the key initiative of this? So the key initiative is Mera Gaon Meri Dharohar. Okay, launched in June 2023 to document the cultural heritage of 6